So I want to introduce Dr. Bill Gibson. So Dr. Gibson is a clinical geneticist with an interest in overgrowth, severe obesity, and lipodystrophy. Ultimately, the goal of his lab is to define therapies for rare disorders like TBRS, and then hope to translate these disorders into viable treatments for prevention of obesity, type 2 diabetes, brain aneurysms, and cardiovascular disease in the population as a whole. So what we're going to do is we're going to do these next sessions for the rest of the day in sort of panels. So Dr. Gibson and the next speaker, Dr. Awamleh, are going to each give their talks, and then we're going to field questions to each of them together for 20 minutes, okay? So without further ado, I'm happy to introduce Dr. Gibson.
Thanks very much, Eric. So just while we're getting the slides set up, the slides are relatively complex, and I apologize for that. If you like complex slides, pay attention to the slides. If you don't like complex slides, pay attention to my words. I'll do my best to simplify things. It's a great pleasure to be here at the TBRS Summit and to be able to talk about a few of the things that have been coming our way at the PRC2 complex registry. So my own lab runs a registry for patients with rare disorders of the PRC2 complex, including Weaver syndrome, Cohen-Gibson syndrome, and SUS12-related neurodevelopmental disorder, sometimes called Imagawa-Matsumoto syndrome. Now, as part of this, we have run into some, shall we say, anecdotal evidence of obesity and other disorders that are related to genetic overgrowth syndromes. And we do have a few patients whom we follow who have let us know about that. And one of the things that we try to do is figure out, well, what should we do with this anecdotal information that we have? How can we best get that out there? And that's one of the things that I'd like to mention. It can be a challenge in these kinds of disorders when you hear about something but it's not formally part of a clinical trial. It's become common practice among Canadian academics to offer a land acknowledgement at the beginning of the talk. I won't read it out because I read it out yesterday, but please feel free to approach me afterwards. And so here we go. I do have a few disclosures in the sense that I will be mentioning some existing drugs that are on the market. I won't be endorsing any particular ones. So just to let you know about that, again, feel free to chat with me about it afterwards. So one of the things that we know about human body size and human body composition is that there are rare genetic variants that have a large effect on things like height, body weight, and head circumference. Of course, Marfan syndrome and achondroplasia are two of the better known rare genetic disorders there. Congenital leptin deficiency is one of the known obesity disorders that sort of where I got my start in rare genetic disorder therapies. This is a patient from Calgary whom we treated with leptin back in 1997, and it had quite dramatic effects on her body weight. So this is data from one of Kate's early papers showing the prevalence of obesity in patients with TBRS, which she's already presented and talked about here. And of course, we know that there are now a number of different mouse models of TBRS that also feature obesity. One of the things that is interesting to note is that the three different mouse models actually have different what we call phagia phenotypes. In other words, some eat more than average, others seem to eat about the same as their age-matched littermates, and others eat less than average. So we've got a nice allelic series here among the mouse models. So there are a variety of different things that control someone's body weight, including the interaction between genes and environment. Most body weight genes are not yet known, and most of the ones that are known have a very small effect size. Nevertheless, rare genes are very important in terms of the contribution to body weight, and it does seem that the downstream effects of DNMT3A mutations can have large effects on body weight. This has also been shown in some other rare genetic disorders. Interestingly enough, patients with mutations in a different gene called P10 seem to be predisposed to obesity, but with a lower prevalence of diabetes. So there's a lot of interesting biology that's going on there. Now, one of the things that is known, of course, is that leptin is a hormone made by fat cells that signals to the brain, in a part of the brain at the bottom called the hypothalamus, how much fat the body has, and that's a very important regulator of body fat. This is a diagram of some of the neural connections in the hypothalamus. This is actually from my PhD thesis, so I trot it out as much as I can because it took me a long time to make it back in the day. But anyway, the thing to know is that there are leptin-sensitive neurons that then connect to downstream what are called second-order neurons that help convert this signal into something the body can understand. So key messages here are that the brainstem receptors, as well as the receptors in the hypothalamus, help the brain understand how much fat the body has. Now, what we have here are some images of one of my patients who has a mutation in EZH2, who has Weaver syndrome, who has miswiring in part of his brain called polymicrogyria. One of my other patients who has mosaicism for what we think is a TBRS mutation also has polymicrogyria on both sides of her brain. Here's an image from someone without polymicrogyria, just so you could see some of the differences. It's the smaller and thicker gyri there. And so any of these mutations that affect the way the brain wires itself can either sometimes cause internal congenital anomalies of the brain, or at least in principle, can change the wiring of the way the hypothalamus is connected. The emerging data from some of these patients and the mouse models suggests that there may be ways to modulate body weight in TBRS. So what I'm showing here is one of the key receptors called the melanocortin-4 receptor that's expressed on the hypothalamus. Basically, just think of it like a long squiggly molecule that binds to a floaty bit, which is the hormone. In this case, it's a hormone called alpha MSH. And it is the target of one of the drugs that is coming down the pipeline called setmelanotide. Now, what I'm showing here is a bunch of different rare variants in this MC4R receptor, some of which have been associated with obesity up here, and some of which have actually been associated with thinness. This is some work done by the group I used to work with at the University of Cambridge. Point being that the shape of proteins is very important to how they work, and it does seem to be one of the master switches for how the body regulates its body weight. So the key thing to know is that different rare variants in that particular protein help modulate body weight. We do think that different variants in DNMT3A may also have different effects on body weight. It's hard to tell without more data, and hopefully the registry will ultimately show us the way there. So one of the things I sometimes bring out to help people understand how we think about these things is this famous quote from Donald Rumsfeld about the known knowns, the known unknowns, and the unknown unknowns. I think those are all sort of people have a good idea of what all of that means. I'm quite interested in this fourth category called the unknown knowns, which is kind of a weird thing to think about. Like, how could you know something but not know it? And essentially, one of the definitions that I've encountered for this is that the unknown knowns are these kind of silent presuppositions that we're not really aware of. Another way to think about it is the things that we sort of know but that we don't actively think about. And so one of the challenges, of course, one of the unknown knowns in rare diseases are, well, can we repurpose some of these existing drugs that have been had safety proven in either common diseases or other rare diseases, and is that something that's possible to do? Often, though, this happens with kind of individual off-label use, and that makes it, at least in my view, an unknown known. It would be useful to know the outcomes of those studies, but they're often not reported anywhere, and we often don't find out about them. So this is my plea to if anyone is actually on one of these off-label drugs, to at least put that information in the registry so that at least it becomes somewhat accessible. What we prefer to do, of course, is have proper clinical trials of drugs, even N of 1 clinical trials. And there's also the possibility of setting up proper trials that have not just one patient, but a few patients who are enrolled with specific monitoring of the drugs. Now, I know I'm probably getting close to the end of my time there, so I'll just mention, you may hear about these drugs, you may hear about things like leptin, you may hear about things like setmelanotide. Neither of these have been proven in TBRS, and it's important to be careful about shall we say, becoming enamored of the next, latest, coolest thing that seems to be coming down the pipeline, because sometimes these things have off-target effects, sometimes their efficacy is somewhat dubious. The patient whose brain I showed you with the EZH2 mutation, he's actually been on Ozempic, so one of the GLP-1 agonists for some time. With the anecdotal reports from the family, it did seem to be helping control his hyperphagia, but more recently they've said it seems to be losing its efficacy in him. So it is very difficult to know what's the best thing to do until you have more data from that. It would certainly be useful for us to have data from some of the mouse models. These are excellent ways to test preclinical therapies. So with that, I'll close out. I'd be happy to take questions from the audience. And thanks very much, everyone, for your attention.
Okay, up next, I would like to introduce Dr. Zain Awamleh. Dr. Awamleh is a postdoctoral research fellow in Dr. Rosanna Wexberg's lab at the Hospital for Sick Children in Toronto. Dr. Awamleh received her honors bachelor degree specializing in genetics in 2013. She then earned her doctoral degree from Western University in London, Ontario, as did I, in 2019 in biochemistry and developmental biology. She then has been pursuing her postdoctoral research at Sick Kids, investigating methylation associated with rare neurodevelopmental disorders such as TBRS, and she's going to be sharing that with us today.
Fantastic. Good afternoon, everyone. Sorry about that. Thank you, Eric, for the introduction, and thank you to the TBRS community for hosting such a fantastic event. It's really a pleasure and honor to be amongst the families. I talked about this at the virtual meeting last year, so if some of you have seen it, hopefully this is a good refresher for you, and also hoping to give some updates towards the end as to where we are. So Dr. Tatton-Brown and Dr. Goodell really helped me out by introducing some basic concepts on genetics, so I'm really hoping to build further on that. So I want to talk a little bit more about epigenetics. So Dr. Tatton-Brown kind of described to you this double helix structure that wraps around itself. Now, that is actually 3.2 billion base pairs long. And so the question is, how do that many base pairs compact and fit into each of our cells? And that's really where epigenetics comes in. It's the mechanisms that organize and regulate and compact our DNA into every cell. Epi is actually Latin for above, and so you can think of it as kind of a higher organizational level of our genome. So this is a schematic that shows you the double helix structure coiling around itself, and then it further compacts by wrapping around these globular proteins called histones. And the way the regulation happens is by the deposition of chemical marks. And the chemical marks can either be directly deposited on the DNA. Can you actually see my cursor or no? Yes? No? No? All right, so chemical mark is deposited right on the DNA, or they could also be deposited on the exposed tails of these globular proteins that we call histones. And it's really the collection of marks at a specific site, both on the histones, on the proteins, and on the DNA that determine the state that the chromatin is in. And it determines whether it's compacted or whether it's decompacted. And that determines whether gene expression takes place or whether that message is translated into a protein. And so these marks don't really act in silo. They communicate with one another, and they send parallel messages in each cell to express or translate certain messages based on that cell type or that tissue. In our work, we are really interested in looking at DNA methylation. So DNA methylation is the addition of a methyl group directly to the DNA. Methyl group is a carbon and 3 hydrogens. The structure is up at the top right-hand side of the screen. And we know that this methylation occurs at specific sites throughout the genome. So where it happens is actually known and well mapped. The other advantage we have is that it's highly stable in cells and tissues, and so we're able to assay it and measure it without being concerned about its degradation. And of course, we know that there is a relationship between DNA methylation and gene expression that, like I had described earlier, could really determine the state of the chromatin. So how do we measure DNA methylation? We do it using a microarray chip. This chip kind of has all these sites that we know can be methylated in the genome mapped out on it. It's produced by a company called Illumina. The current version, or the version that we had been using, version 1, maps out more than 850,000 sites across the genome, sites where methylation could happen. And so we, at specific sites, we would take a cohort of individuals with neurodevelopmental disorders and a cohort of individuals that are considered typically developing, and we compare the methylation. And so at many sites, the methylation is similar. What we're really interested in is the sites where the methylation is really different between the typically developing individuals and individuals with neurodevelopmental disorders. Once we identify what's different, it often results in a pattern, and we start to refer to them as DNA methylation signatures. So that is really just indicating that there's a robust pattern that we have quantified of DNA methylation differences between the two groups. So Dr. Wexberg has been has been a pioneer in this area, and in 2013, her research group was the first to identify that there are specific patterns of DNA methylation that are of very large effect size that are found in individuals with neurodevelopmental disorders. One of the first was KDM5C in Class-Jensen syndrome, and subsequently we also identified DNA methylation changes in NSD1 in Soto syndrome and KMT2D in Kabuki syndrome. To date, there's probably more than 70 genes that are known to cause these neurodevelopmental disorders. And this has come up quite a bit yesterday and today. The big challenge is interpreting those sequence changes, and particularly ones that are classified as variants of uncertain significance. So we start to think about how we can use these signatures to perhaps help us and be a diagnostic tool. Because they're so specific to the disorders and they're so sensitive, could we use them to help us interpret some of those variants of uncertain significance? One thing I want to point out is that all of these genes, those more than 70 genes that I mentioned, they really all function in maintaining the epigenome. So that could be in depositing a mark on a histone, reading a mark, removing a mark. And so there's this commonality across all of these genes that they function in epigenetics, and detrimental sequence changes in them are associated with neurodevelopmental disorders. And so in the context of TBRS, we start to think about how DNA methylation changes in TBRS can help support some of our research goals, particularly trying to explore DNA methylation as a diagnostic biomarker that can complement other diagnostic tools to help us interpret those VUSs. And so we had a preliminary cohort of individuals. We typically use whole blood-derived DNA. We use the EPIC V1 array that I described to you that assays more than 850,000 sites. we had 9 individuals, and we split that into 2 groups, 6 individuals in the discovery cohort, and set aside 3 individuals for independent validation. And we also ensured that we have enough controls that we can detect a difference. So we have typically developing sex and age matched controls. You've probably seen this schematic or some sort of form of the schematic a few times. So it's the DNMT3A gene, and plotted on it are all the individuals that are in this cohort. In addition to the 9, we had 2 that had variants of uncertain significance. And so each lollipop is an individual, and you can see that we have a mix of frame shifts and missense and some splice variants as well. So with these 6 samples and the 17 controls, and in accounting for sex and age and some of the cell types, we identified a pattern that's made up of about 1,100 sites. Those sites were selected with strict statistical thresholds. So the adjusted p-value is less than 0.05, and the effect size is greater than 20%. We can visualize this is called a principal component analysis plot, and essentially it's a plot that just helps deconstruct your data and show you where the largest differences are between your group. And so kind of deconstructs your data into this 3D plot. And what we see here is that based on that pattern we identified, based on approximately 1,100 sites, that's really separating the control individuals from the individuals with the pathogenic DNMT3A variants. And so now that we have this pattern, we wanted to put it to the test. Of course, we wanted to check it in the 3 individuals that we set aside, and we also want to compare it to perhaps some of the other DNA methylation profiles of individuals with other neurodevelopmental disorders. So how do we put it to the test? We use a machine learning model. We train the model based on the discovery controls as well as the discovery cases, and we ask this model to provide us with a probability between 0 and 1 for every subsequent DNA methylation profile we give it. The closer that probability is to 1, the more likely that individual has a DNA methylation profile that's similar to individuals with TBRS. The lower the probability closer to 0, that indicates that their DNA methylation profile is more similar to control individuals. So first, we wanted to include 100 controls of a mix of ages and sexes and ethnicities to score. And you can see that our model scored them all well below 25%, indicating that our model is doing a good job distinguishing the 2 groups. We next scored the 3 individuals that we set aside for validation, and you can see that they all scored well above 75%, confirming that our model is working. And then lastly, we also compared or included in the model DNA methylation profiles of individuals with Kabuki and Weaver syndrome that are caused by pathogenic variants in KMT2D and EZH2. And what you can see is that they all have scores well below 25%, similar to the controls. So this indicates that this pattern is really specific to TBRS, and we're not seeing it in other neurodevelopmental disorders. So now that we're more confident in this signature and in our pattern and our model, we can look at classifying the 2 VUSs that I had mentioned earlier. And so you can see here the 2 cases that were originally variants of uncertain significance actually have DNA methylation profiles well above 75%, indicating that they likely do have TBRS, and this was in line with the phenotype that they had described, which was also TBRS-like. So, this is my slide from last year, because I kind of wanted to show you what I put up for my goals last year and where I'm at now. We really wanted to work on collecting additional samples and expanding the signature, because this is really our preliminary data. We wanted to collect more variants across the entire gene and in different domains, and of course, knowing its diagnostic utility. The biggest thing is really reaching out to the community and identifying those VUSs so that we can score them. So what have we done in the last year? In terms of, we were successful in applying for funding with the TBRS community and uplifting athletes, so we received funds to expand the signature, which is fantastic. We have collected a lot more samples, so my map looks different from the first one. We now have 29 samples, in addition to the 11 that we previously had. And you can see we now, although missense is still the most prominent variant type, you can see that we have variants spread out across the gene. We kind of waited a little bit because Illumina, the array company, upgraded their platform. So they've identified more sites in our genome where methylation changes. And so we decided to hold the cohort so we can put it on the V2 array. So this one assays more than 935,000. So they identified approximately an extra 100,000 sites that are important. So I'm happy to report that all of these samples are currently being processed at the array facility, so I hopefully should be getting data within the next month. So I really expect that we will have this new expanded signature before the end of the year, and I can't wait to report on it back to you all. Another thing we started looking at is the Haynes-Sproul-Jackson syndrome. So in 2019, there's a paper from Dr. Andrew Jackson and Dr. Sproul, Duncan Sproul, from the University of Edinburgh, that perhaps where they found a cohort of individuals, I believe it was 5 reported in this paper, that had variants in DNMT3A, but actually had the opposite phenotype to TBRS patients. They did have developmental delay and intellectual disability. However, they had short stature and decreased weight, and some of them also had intrauterine growth restriction. So an undergrowth phenotype compared to the overgrowth that we see in TBRS. So I reached out to Dr. Jackson. And I said, hey, let's collaborate. Let's get some samples. Let's do something about this. And interestingly enough, he had already run about 7 samples. So at the top, you see all the cohort that we have. And in the bottom, you can see the 7 individuals with Haynes-Sproul-Jackson. And he had kind of done a preliminary study and run them on the EPIC V1, similar to the platform that we used in our preliminary study as well. You can see they're all concentrated in this PWWP domain. And so we thought, why not take the profiles and just cluster them on the principal component analysis plot and see what they look like. So interestingly enough, I'll say the Haynes-Sproul-Jackson samples here are in blue. Our TBRS cohort that I just presented to you on is in the variations of red. And then in the controls, you have our own internal controls, as well as controls that were run in Dr. Jackson's lab along with those cases. And what we see is this separation between the controls and the individuals that have variants in DNMT3A. But the Haynes-Sproul-Jackson samples are kind of separated from the TBRS samples. They're not fully overlapping them. So we're hypothesizing that there is likely a unique but overlapping signal for the Haynes-Sproul-Jackson syndrome. And so in collaboration with Dr. Jackson, we're currently working on collecting additional samples and developing a signature for this syndrome and really compare the two that we find between TBRS and Haynes-Sproul-Jackson. So now my current goals, what I'm going to do for the next year. We're going to, like I mentioned, complete the development of the expanded signature and hopefully have it by the end of the year. We really want to take a closer look perhaps at some of the correlations between the genotypes, particularly the variant types, as well as the phenotype, and if we see some correlation to the DNA methylation changes. And some of the subphenotypes we're interested in are things like ASD, psychosis, AML at the arginine 882 variant. Hopefully also in the next year, like I said, we would like to develop a signature for Haynes-Sproul-Jackson and make that comparison between the two cohorts and really better understand what is overlapping and what's unique between the two. And ultimately, we're trying to work towards using the DNA methylation signature as potentially a functional biomarker for therapeutics. So it can give us an output as to whether a particular therapy is working or not. And that's really our goal in the next few years. I really want to take the time to thank the community. Without you, this work is not possible. I'm just really get speechless and tongue-tied about it because I'm just so overwhelmed by how amazing you all are. So keep doing what you're doing. Let's keep supporting each other. Go out there, donate samples, participate in research because it really is impactful. With that, I want to thank all our collaborators and all of you for listening, and I'm happy to take any questions. Thank you.